Hello, and thank you for joining us. I'm Gwen Taylor, Senior Developmental Editor and Webinar Program Manager with Current Protocols at John Wiley & Sons, and I'm delighted to introduce today's webinar titled Choosing Labels and Conjugating Antibodies for Multicolor Flow Cytometry and Other Immunoassays. This webinar is being co-sponsored by Current Protocols and MilliporeSigma. MilliporeSigma is the North American life sciences business of Merck KGaA Darmstadt, Germany, and is dedicated to providing scientists and engineers with best-in-class technologies, lab materials, and services with the intention of making research and production simpler, faster, and more successful. The company is deeply committed to providing novel cell culture systems and characterization tools for stem cell research and primary cell culture, and offers a comprehensive line of stem cell products, including human and murine stem cells, media optimized for stem cell and primary cell culture, growth factors, exclusive antibodies, cultureware, and kits to help you jumpstart your research. Current Protocols is in its 29th year and is the largest collection of peer-reviewed, regularly updated step-by-step research protocols available for life scientists worldwide. With 18 titles and over 17,000 protocols, Current Protocols is part of Wiley Publishers. During today's program, we encourage you to submit your questions throughout the webinar by clicking on the Ask a Question box at the bottom of your screen. Your questions will not be seen by any of the other attendees. The webinar will be recorded and available for viewing in the next few days, and we will send you an email with details on how to access the on-demand webinar, along with a customizable certificate of attendance and a PDF of the slides. So now it's my pleasure to introduce today's speakers. Dr. Timothy Bushnell is the director of the University of Rochester Medical Center Shared Research Laboratories, in which he is responsible for the management and oversight of 14 research core facilities. He is also the scientific and technical director of the University of Rochester Flow Cytometry Facility, as well as a co-founder of Expert Cytometry, a flow cytometry consulting and training company. He earned his PhD in biology from Rensselaer Polytechnic Institute and did postdoctoral research at the University of Rochester Medical Center with Dr. Fei Young on early B-cell development. Dr. Bushnell's current main focus at Rochester is the development and application of high-end flow cytometric assays to support the research programs of members of the University of Rochester community. In addition, Dr. Bushnell has been involved in outreach as a member of the ISAC Council since 2008, and he founded the Western New York Flow Users Group. Our second speaker, Dr. Amedeo Cappione, has over 20 years' experience in molecular and cellular biology, specializing in the development of cell and protein-based assays. Dr. Cappione obtained his PhD from the University of Rochester Medical Center, exploring changes in post-transcriptional RNA processing in neural cancer. As a postdoctoral fellow in the lab of Dr. Ignacio Sanz, Dr. Cappione's work focused on understanding the mechanisms underlying loss of B-cell tolerance in lupus disease. As a member of MilliporeSigma's applications group, his current focus is primarily on the development of products to streamline cell culture and protein sample preparation workflows. So let's go ahead and get started with a very warm welcome to you, Dr. Bushnell.
Thank you, Gwen, and thank you, Wiley and Millipore, for giving me this opportunity to present and discuss principles and practices of polychromatic panel design. My email is here if you have additional questions after the webinar. I'd be more than happy to answer those for you. But look forward to an engaging discussion after this presentation. The educational objectives I hope to achieve today is that you will understand the principles of designing polychromatic flow panels and how to integrate these into your current workflows. The principles that we will cover today include understanding how to optimize a flow cytometer to determine the relative error of contributed to different detectors, how to pair fluorochrome brightness with antigen density, the steps for optimizing a polychromatic flow panel, and last but not least, where to get more help. The first question you can ask is, why polychromatic flow cytometry? Well, more colors means that we have more populations. It helps us improve the resolution of subsets of interest. From the early days of flow cytometry, where we could do 3 and 4 fluorescent markers, to tools now that let us do 15, 18, as many as 30 markers, we're able to really ask very detailed questions at the cellular level. These more colors also lead to these more questions. We're able to phenotypically define questions and answers, looking at rare event analysis such as stem cells, rare events being produced because of disease states or treatments, all of which require this high-end polychromatic flow. Of course, with polychromatic flow, more colors also means more problems. As fluorescent tags increase, there's a potential for loss of resolution in the markers and in the channels of interest. So we need to take that into account as we build and develop our polychromatic flow panels. Step 1, focus on the experimental question. By focusing on the experimental question, it helps us define the populations of interest. What antigens we will need to define the cells that we are studying in our assay. Once we've defined the cells we're studying, we need to focus on the data that we need to extract from our experiment. This data could be a change in population. For example, an increase in a population, say CD4 T cells, in a patient treated with a new drug. Or it could be a change in expression, say an increase in an activation marker like CD69 on a specific subset of cells. Knowing what data we need to extract lets us define what statistical analysis tests will be performed, and it's really important to define that protocol before you start your experiments so that you know going in how much data you need to collect, how many experiments you need to run, and how you plan to analyze that data. The second step in polychromatic panel design is your instrument choice. You want to select your instrument with care from what's available in your local institution. The excitation lines on the instrument will dictate what fluorochromes you can use in your polychromatic panel. The emission filters on your instrument will dictate, 1, where a given fluorescence signal can be measured, and 2, where a given fluorescence might cause issues, and we'll illustrate those in a minute. Last but not least, you want to know that this instrument has been optimized, so you have an understanding of how the machine behaves as you design your panel, and design the panel to make best use of that instrument, where it's most sensitive and where it's least sensitive. Laser wavelength matters. Here's an example looking at phycoerythrin, PE, a commonly used fluorochrome. Many instruments have a 488 nanometer excitation laser. Some, as shown in this case, have a 532 excitation laser. You can see from the excitation and emission profile on the right that if you use the 488 nanometer laser, you get about 60% excitation efficiency. The 532 shows about 80% excitation efficiency. This translates into a brighter signal with the same laser power as shown on the panel on the left, where we have 2 sets of cells stained with a PE-labeled antibody, and the fluorescent intensity with the 488 laser is only about 10,000, whereas with the 532 laser, it's about almost 20,000. That difference in excitation source can improve your resolution. So knowing where you're going to measure this and what lasers are available becomes important. Excitation sources, laser power matters. As shown on this slide, when we increase the laser power from 50 milliwatts, shown on the right, to 100 milliwatts, shown on the left, the intensity doubles. More laser power means more energy going to the excitation fluorochrome, and therefore a more energetic emission, translated into brighter signals. Brighter signals help us resolve our populations of interest better. So, knowing what laser lines are there and what their power is very important. The other thing to worry about, as we mentioned, was the emission profiles. Here's an example of an instrument showing the different laser powers of the 4 lasers on this machine and the different detectors, the long pass and the band pass filters. We're going to focus on that band pass filter for the most part because that is directly in front of our photomultiplier tube. That dictates the wavelengths of light that will come into that PMT and therefore what fluorochromes can be detected by that PMT. Interestingly enough, if we focus on the violet laser for a minute, as shown here, we start to see that the effective filter on this instrument are not the filters that you see published, but it is a combination of the filters in front of and behind that PMT, which can reduce the effective range of the filter, therefore changing what fluorochromes can be measured by that filter, as well as what fluorochromes may cause spillover and loss of resolution into that channel. You can see here, for example, the 560/40 bandpass filter here is actually really only a 563/13 filter. So we're only getting about 13 nanometers of wavelength of light through that filter, as opposed to the 40 that we thought we'd have based upon that bandpass filter. Having this information will help you know where your fluorochromes will be measured and where they're going to cause problems. And here's yet another example of those potential problems. Here are some common fluorochromes listed on the left and where they would be measured on this machine of interest in the primary detector. The possible detector issues shown on the far right indicate where that fluorochrome is going to potentially have measurable signal, therefore impacting the sensitivity and detection in that channel. Quantum dot 705, for example, is typically measured in the violet B channel, excited by the 405 nanometer laser and measured by a 710/50 bandpass filter. That 710/50 bandpass filter happens to be on several other detectors, and quantum dots are excited by every wavelength of light below their emission maximum. So you can see Q dot 705 can impact the green, violet, and red lasers as well, affecting 4 channels in total, meaning it's very difficult to design a polychromatic panel around that. Other fluorochromes that might be of interest that people are using, for example, PerCP Sci 5.5. Everyone is used to using this off the 488 laser, but as you'll see, it can impact the violet laser because there's about a 25% excitation of PerCP off of the violet laser. And that is shown here. Recent advances in characterizing instruments have been published, and one such paper is shown here. This was published in 2013, coming out of Mario Roederer's lab. The goal of their characterization is to identify what fluorochromes impact other detectors, reducing sensitivity in that detector. This measurement can therefore predict where you might lose resolution in a channel that you might want to avoid. Now, the details of the math are shown in this paper. The consequences of that are shown here for one instrument. It's a large table. It's very hard to see. I apologize for that. It looks better in the PDF. But if we go across the top of this, we have the different fluorochromes and how much error each fluorochrome contributes to each detector, which are listed down the side. If we sum down the column, we get the amount of error a given fluorochrome will contribute if it is in the panel to all the detectors, therefore the loss of resolution of a given fluorochrome. I'm going to focus for a minute on these 3 pairs of fluorochromes highlighted in, and that highlight is shown here. We'll take them one at a time. For example, 2 common fluorochromes, PE Texas Red and PE CF 594. have similar emissions. However, if we look at the error that each one contributes to a panel, if it was in that panel, you can see PECF594 in this example contributes about 3 times the amount of error as PE Texas Red. Given a choice, this becomes a fluorochrome that I would like to avoid because that error contribution is reducing sensitivity, especially in several channels where I might want to make other sensitive measurements. For example, the blue channel shown at the top, it's about 4 times more error with that CF594. If we take a look at the quantum dots in the middle panel, Q.565 versus brilliant violet 570, we can see that that brilliant violet dye contributes a great deal of error throughout the panel. Again, almost 10 times the amount of error, making it, while bright, another poor choice for panel design. And if you wish to use it, we must design around the errors, around the channels that it causes the most error in. In this case, 2 of the violet channels where it's contributing 1,000 and 1,600 channel values of error. Last but not least, Pacific Blue and Brilliant Violet 421, 2 other fluorochromes. And you can see these are actually about equivalent when it comes to panel design, contributing very little error in either case. What I can do for my panel design is take this information and give you a chart for your instrument that gives you what detectors receive the most error, assuming all the fluorochromes are present in the panel, versus what fluorochrome, shown on the right, contributes the most error. For example, on this instrument, the blue A 488 excitation with a 51050 bandpass filter, which was shown several slides ago, receives almost 10,000 channel values of error from all the fluorochromes listed on the right. PE Sci5 and PE Sci5.5 each contribute a great amount of error into the panel. Therefore, using those would not be good if we wanted to make sensitive measurements, because they're going to cause a great deal of error in a lot of places. And we want to design around using that blue A detector to receive and make sensitive measurements. Now that we've talked about the instrument, let's discuss about antibodies, because the antibodies become our workhorse and are how we measure the how we phenotypically define our cells of interest. And we're going to focus on the specificity of these antibodies. Overall, what we are focused on is the antibody binding to the desired epitope with a very strong K-on reaction, very slow K-off reaction, so it's not falling off our target, has a high affinity. Anytime that antibody binds to a non-target, that will increase background. The 2 of the areas that we have to worry about are Fc binding, so the Fc receptor on certain cell types binding to the Fc fragment of the antibody. This is a specific reaction, but not one we're typically measuring with our antibodies. We can minimize that Fc binding using various blocking reagents, such as normal serum, Fc block antibodies, etc. This reduces that specific binding, which causes a loss of sensitivity. The other area that is important is non-specific binding. This is the idea that the antibodies will bind to anything that is, any protein that is there if they can't find their primary target. This non-specific binding is increased when we have a lot of antibody present. So in addition to blocking, we need to perform titration experiments to find the optimal concentration of antibody that we wish to use in our assays. The other thing that you want to worry about is different types of antibodies to the same target, different clones, they have different affinities. And I found in the case of human antibodies, this website, www.hcdm.org, lists the various approved clones for different antigens and tells you where you can find them and gives you information about them. So this will help you, the investigator, as you're designing the panel, make a choice of the 4 or 5 different CD3s that are available and other antibodies in targets, so you can identify the correct best antigen, best antibody to use. How do we apply this to our panel design? Well, the way I like to think about it is I'm going to classify my targets at 3 levels. Over here on the right, I'm showing a typical developmental diagram you've probably seen of the hematopoietic system from stem cell to mature cells. My primary or major target antibodies, these are the things that are highly expressed. We talk about these cells all the time, things like CD4, CD3, CD19 for B cells in the human, for example. The second level of antibodies that we talk about are our subsetting targets. These are our fine-tuning targets. This helps us refine our population, moving from just a B cell or a T cell down into a activated regulatory T cell or a plasma cell, B cell, for example. These have a variable range of expression and are critical for having good separation so we can really hone in on the cells that we're interested in. The third classification are my tertiary or my unknown targets. These either I have no idea what their expression is, or these are the targets that I'm actually questioning and interested in. So these are the ones that are going to be used to answer my biological questions. Now we're going to put this aside for a minute. We're going to talk about fluorescence because we're going to then couple these together. When we talk about fluorescence, what we're really interested in is how bright is a given fluorochrome. The reason we're interested in brightness is because the brighter a fluorochrome is, the more signal we get out, the easier it is to make a sensitive measurement. There are many antibodies out there, many fluorochromes out there, and the question is, which fluorochrome do I choose? And to study that, several people have proposed what's called the staining index, a metric to rank fluorochromes. And that looks something like this. Here, we measure the distance between the positive sample, the bright fluorochrome, and the negative population. We measure this typically using the median fluorescence intensity, and then we correct for the spread of the data of the negative population. So the effective brightness of a reagent is the difference between the positive and negative divided by the spread of the population. That leads us to be able to create charts that look like this. This is a chart slide from Holden Macker, but there are many charts like this out on the web and should be designed for your specific instrument because every machine out there can have different laser powers, filters, etc. So you want to be able to have it as close to your machine as possible. But what this staining index analysis tells us is it tells us the brightness related to each other. You can see here we have the reagent fluorochrome all the way over on the left, the clone that was used, and the filter combination. We can see things like phycoerythrin as considered one of the brighter fluorochromes, whereas something like Pacific Blue and AmCyan are much less bright, about 10-fold less bright than PE. So if I want to make a sensitive measurement, I want to be at the upper end of this chart, the PE, the APCs, the Alexa 647s, as opposed to the Pacific Blues and AmCyans. Now there's been many fluorochromes added since this chart was made, so I encourage you to use Google and find some charts that have been published to help you with some of the newer fluorochromes, or like I said, do the experiment yourself and rate them on your instrument. Now, I take this information and I go back to how I've classified my antibodies. So my primary targets, my big cellular subsetters, I'm going to use the dim fluorochromes, things that are moderate or dim, Pacific blue, AmCyan. I'm going to trade off the fact that these antibodies are highly expressed on the surface of the cell and use a dim marker to have a reasonable signal. For my subsetting targets, so secondary targets, I want to use a moderately bright fluorochrome that's going to give me good separation. So I'm going to try to find areas on my instrument where I don't have a lot of loss of sensitivity based upon all the other fluorochromes in my panel, so that I can properly find and subset, drill down to those cells of interest. Finally, I'm going to then pair the markers that I'm most interested in, the ones that I'm actually doing the experiment about, with the brightest fluorochromes I possibly can. I want to get the best resolution and the best sensitivity, so brighter is better. This is where I would definitely want to use things like PE, APC, some of the newer brilliant violets, some of the quantum dots for that assay. There are two caveats to panel design that you should take into account. If you're going to be doing intracellular staining, size does matter. Different fluorochromes have different are of different sizes. Things like FITC, fluorescein, are much smaller than PE, which is a protein. And if I'm going to be doing intracellular staining, I want to use smaller fluorochromes because they make it easier to get into the cell than a big antibody like PE or APC. Think about trying to get a big protein already, the antibody, through a small hole that you punctured into the cell with another big honking antibody protein on the end. The second caveat about panel design and fluorochromes is protocols may affect your staining choice. Depending on what your protocol is, especially if you're going to be doing fixation permeabilization, you may have to compromise and make certain choices because some fluorochromes hold up to fixation permeabilization better than others. So those two caveats have to be taken into account when you design your panels. Whenever I've worked with people in the past designing panels, after going through this exercise of identifying what antigens are available, it used to be send them out with a worksheet to pull out what antibody fluorochrome combinations there are out there in the commercial market. And that could take several days or weeks, depending on how complicated their panel was. There are several tools out there now that help improve automation and panel design. What all of these tools do, Flourish, Chromocyte, and Fluorofinder, is they serve as a way to search the databases in a systematic fashion along with the characteristics of your instrument so that you can choose antibody fluorochrome combinations, one, that are commercially available, and two, that will work on your instrument. And if you haven't tried one of these programs, I would encourage you to take advantage of them. All three are free for academics. If you're a commercial entity, you'll have to talk to the different companies to find out how they work that. But I have found that these programs save me countless hours of searching through databases and really speed up the process of panel design in an automated fashion. All of these programs work with the same. They allow you to input the instrument that you're using. This includes the excitation lines and the emission filters. So the software then knows what fluorochromes are possible and where there's going to be overlap, and will often give you warnings as to saying if you choose this fluorochrome, it's going to affect these other channels. It lets you input the target of interest. This could be, this is not only the target species you're working with, human, mouse, rat, etc., but the specific antibodies you're interested in. At that point, it searches those databases. It searches all the databases that each program has available and then provides you with the list of fluorochrome antibody combinations that will work on your machine, answering the question you want. You then must manually select the antigen fluorochrome combinations you want. And this is where knowing the sensitivity of your instrument and the brightness of the fluorochrome will be helpful for you to make those choices. There is no automated panel design as of yet, but this is something that people keep trying to push and develop, and at some point this may become an automated process. But for now, it's you, the researcher, taking advantage of understanding your instrument, understanding brightness, and the question you want to ask to pick and choose the right fluorochromes. Now, don't be concerned or frustrated if you choose a panel and you run it and there's issues. You sometimes have to go through in an iterative process and tweak out one or two of the fluorochromes antibody combinations. We've developed panels here that have taken as long as 15 months to get optimized because we have, when you do high-end polychromatic fluor, you're making these trade-offs. Now, that seems like a lot of work. It can be a lot of work. So there is a shortcut, a solution for it. It's called the OMIP, the Optimized Multicolor Immunofluorescent Panel. It was a journal type that recently came out, recently I say 2010. The beauty of the OMIP is that it is a way for you to find panels that have been developed and optimized. These panels include a summary table of what the panel is for and what reagents were used, all of which must be commercially available reagents so that you know that you can get access to the antigens of choice in this panel. The other nice thing about the OMIP, which there are 31 of them published right now, and you can get a link to them all here. These are the first 4, is this feature right here. It shows you a gating strategy that the researchers who used and developed this OMIP used to identify their cells of interest. This is from OMIP 01, and you can see here the summary table, what reagents were used, what the fluorochrome choice was and the purpose, and then their analysis so that you have a guide to help you in your analysis as well as knowing what fluorochromes they used. And you can see if these will work on your instrument or if not, what equivalent substitutes you can make. The beauty of the OMIPs is that they're an ongoing, evolving process, and you can see OMIPs referring to previous OMIPs so that you can see how people's thought processes are as they evolve to answer specific questions using backbone pieces from one OMIP or another. So I'd like to summarize that this process all starts with the experimental question. You need to know what you're doing. You need to know your statistical downstream applications so that you can then go forward and develop your panel. You need to know your instrument, and this includes the configuration, the excitation and emission profiles of the machine, where the laser lines are and what filters are in front of each detector so you know where the fluorochromes will be measured. You need to know the sensitivity of the machine, how sensitive each PMT is, and more importantly, where there are regions of high or low sensitivity so that you can design your panel around those, taking advantage of regions of high sensitivity and using regions of low sensitivity for things like viability dyes to eliminate dead cells, dump channels to eliminate cells that you're not interested in. You need to know your reagents. The fluorochromes, we need to know things like brightness and their sensitivity to fixation and other chemical conditions that you will affect and introduce the cells with. With the antibodies, we need to know the specificity and the antigen density. Again, we pair our bright fluorochromes with low antigen density targets and our dimmer fluorochromes with our higher energy, higher density targets. And in the end, don't forget your allies. These automated panel design tools, Fluorofinder, Flourish, and Chromocyte, which can help cut through the time it takes to identify the available reagents and helps you visualize which ones are going to work on your machine. And these OMIPs, if in doubt, you can find what may be useful for you in answering your question using the OMIPs. It's reducing the time it takes for you to get your panel up and running. With that, I'd like to thank you and be happy to answer any questions at the end of the webinar. And I will now turn it back over to Gwen and our next speaker.
All right, thank you so much, Dr. Bushnell. So we now proceed to Dr. Amedeo Cappione of MilliporeSigma, who will present work highlighting the use of the Amicon Pro device for small molecule antibody labeling.
Thanks, Gwen. The work I'll be presenting today was originally published in the Journal of Immunological Methods. To start, let's go back and take a look at why the Amicon Pro device was originally developed. The protein purification workflow is hampered by numerous pain points, including tedious multi-step processes requiring multiple devices with the potential for sample loss. In fact, looking on the left, current affinity-based purification schemes require multiple tools to derive a final protein sample in the correct context for downstream applications. The Amicon Pro device is an adaptable centrifugal device coupling resin-based affinity purification with downstream sample concentration and buffer exchange. By condensing the protein preparation workflow into one device, the Amicon Pro eliminates the need for multiple sample transfers, thereby minimizing protein loss. This capability was made possible through incorporation of the Amicon Ultra 0.5 ml filter into the larger Amicon Pro device. Now let's look specifically at how and why the Amicon Pro is well suited for small-scale antibody labeling applications. Protein immunodetection encompasses a broad range of analytical methodologies, including Western blotting, flow cytometry, and microscope-based applications. These assays are relied upon fluorescent or chemically tagged target-specific antibodies. Commercially tagged antibodies are available but are costly, and not every color is available. Kits for labeling exist as well, but these possess issues which are barriers to researchers doing their own antibody labeling. First, the process can be quite long, involving, in certain cases, 2 overnight dialysis steps. Because it's long, there's a tendency to take shortcuts. Researchers are less likely to optimize the labeling reaction through identifying the correct dye-protein molar ratios for their application. There's also significant sample loss, which can be costly when commercial antibodies are to be used. Depicted across the bottom are workflow schematics for antibody labeling using the Amicon Pro device. For concentrated antibodies, greater than 1 mg per ml, the antibody can be added directly to the 0.5 ml filter and then assembled into the device. For dilute antibodies, an initial concentration step can be performed. If the antibody is impure, such as a hybridoma, or contains BSA or other carriers, the device's resin-based affinity purification capability can be exploited for cleanup. Once the antibody has been eluted or concentrated in the 0.5 ml filter, labeling mix is added to the upper exchange reservoir and the reaction initiated by centrifugal diafiltration. Unbound dye is cleared by buffer exchange and the sample is concentrated. This two-step labeling protocol is contained within the dotted red box. The conjugated antibody is recovered from the 0.5 ml filter by reverse spin in a microcentrifuge. As mentioned, one major deterrent to protein labeling faced by researchers is the tedious and inefficient buffer exchange methodologies available. By virtue of its ability to combine buffer exchange with concentration, diafiltration potentially offers the better option for buffer exchange. To maximize the buffer exchange capacity, the base of the Amicon Pro device was designed to exactly match contours of the 0.5 ml Amicon Ultra filter, facilitating optimal exposure of the membrane surface area while reducing volume between the plastic sidewalls. The tip also narrows, promoting sample diafiltration within the exchange chamber. Under continuous centrifugal pressure, buffer is slowly metered in, mixed with the sample, and forced across the membrane boundaries. While many smaller devices have volume constraints and thus require multiple cycles for effective desalting or buffer exchange, this upper reservoir holds up to 10 mL of buffer, permitting a single spin exchange. Depicted at the bottom are the basic steps for buffer exchange and concentration in the Amicon Pro. This process is as simple as adding sample and spinning to concentrate, adding new buffer to buffer exchange and concentrate, and then recovering the sample by reverse spin. In the end, There are many ways to exchange buffer: dialysis, gravity-based desalting columns, and diafiltration. One key point is that both dialysis and gravity-based methods will require use of a second device for sample concentration. To assess the relative capacity for buffer exchange, testing was carried out against representative devices for dialysis, gravity gel filtration, and centrifugal diafiltration. 4 parameters were measured: processing time, buffer exchange efficiency, protein retention, and specific activity, measured as a correlate of functional integrity. The results are summarized here. Overall, the Amicon Pro device offered roughly equivalent or superior performance for all 4 attributes, suggesting it is well suited for antibody labeling. Specifically, highly effective desalting was achieved with 1 15-minute spin using a small volume of buffer. Most devices showed residual sodium chloride levels, suggesting that either there are limits to their exchange capacity or that further processing steps were required. The device demonstrated the highest protein recovery. On average, less than 3% of starting BSA was lost. In fact, all centrifugal-based methods where concentration and buffer exchange are combined had greater protein retention. The device also delivered a protein sample with similar specific activity as the gold standard dialysis method. Lastly, the process of buffer exchange in the Amicon Pro device was relatively fast and required little hands-on effort. Other methods were much longer and required multiple repetitive processing steps. Commercial antibodies are routinely packaged with up to a mg per mil of BSA as stabilizer. The potential impact of BSA on labeling is illustrated here. 50 labeled anti-GAPDH conjugates were used to probe an A431 lysate dilution series. Even at 1 mg per mil, even at 1 microgram per mil, BSA caused a significant reduction in detection sensitivity in Western blotting. This finding is in conflict with the measured FP values for each conjugate shown to the right of each blot panel. At higher BSA levels, the increase in FP suggests that the carrier protein is actually acting as a sink during the reaction. Alternatively, over-labeling may cause a reduction in the probe's binding efficiency. Labeled BSA can also lead to increased background signal. While no such issues were observed with this application, this effect may be lysate or antibody dependent. Because the Amicon Pro device was designed for resin-based affinity purification, general antibody cleanup, including carrier removal, can be easily achieved prior to labeling as previously outlined. The degree of labeling is influenced by molar ratio of reactants, activity of labeling reagents, and the nature of the probes themselves. Optimization is often required to ensure a probe's signal strength is consistent with signal-to-noise requirements of the assay. For fluorescence, a high degree of labeling per antibody is preferred to maximize sensitivity. This is particularly true for multiplex assays where direct conjugates are to be used. However, over-labeling can cause loss of specificity, decreased avidity, and issues with solubility. Fluorophores attached in close proximity can also decrease fluorescence via quenching mechanisms. On the other hand, ineffective labeling will lead to weak fluorescent signals. In the Amicon Pro device, reactions are initiated by buffer exchange with the labeling mix replacing the antibody storage buffer in the 0.5 mil attachment. Because of the centrifugal format, it is possible to assess labeling efficiencies across a range of reaction conditions by quickly halting the reaction through diafiltration-based buffer exchange. Using this method, the FP ratio was determined following labeling in the presence of increasing amounts of NHS fluorescein dye. Bars represent the average FP ratio for an anti-GAPDH antibody labeled with increasing amounts of dye using the Amicon Pro device, or using the standard 2-hour reaction protocol. For the device, 50 micrograms of antibody was loaded directly into the 0.5 mil filter and assembled. The reaction was initiated by buffer exchange and concentration of the labeling mix. Immediately following centrifugation, the sample was buffer exchanged and assayed to determine the probe's FP. Not surprising, the probe's FP values increased with increasing dye input. However, this response appeared to plateau at 30 micrograms, quantity which was 12-fold greater than the kit's recommendations. By contrast, when similar conditions were used following the kit's static 2-hour tube format, FP values did not plateau but continued to increase with dye concentration. The increase was not linear with dye input, suggesting that quenching may occur at higher labeling densities. Immunoblotting with resulting conjugates demonstrated greater signal intensity for the tube-derived antibodies in pairwise comparisons, but also increased background. The relative increase in signal was not proportional to the differences in FP values, indicating a potential loss in binding efficiency among the over-labeled conjugates. These findings are consistent with prior studies that suggest commercial labeling kits stipulate suboptimal fluorophore levels to avoid issues associated with over-labeling. At comparable dye load, the observed differences in FP values between the two methods is potentially best explained by the fact that the centrifugal process is continuous, and thus unbound dye is rapidly forced out of the reaction chamber by buffer exchange, thus minimizing exposure to excess label. A more significant finding was that labeling in the Amicon Pro device was almost instantaneous. Here, labeling kinetics were determined under different conditions using 50 micrograms of anti-GAPDH antibody and NHS fluorescein dye. For the Amicon Pro device, reactions containing 2.5 to 60 micrograms dye were assessed. For the standard reaction performed in a 1.5 ml tube, labeling was carried out using the standard dye input in a 50 microliter reaction volume. Reactions were stopped at the indicated time points by buffer exchange, and an aliquot of the labeled antibody was measured to determine the FP ratio. By contrast to the Amicon Pro, the standard tube reaction resulted in only a small increase, roughly 15%, in the probe's FP when it's assayed immediately following mixing. The degree of labeling was further measured at 15-minute intervals for a 1-hour time course. For each dye level assay in the Amicon Pro device, the maximum FP was reached in 30 minutes. However, approximately 75% of the labeling was achieved by time zero, which would be just after initial centrifugation. Overall, labeling efficiency was more strongly influenced by dye input than reaction time. Consistent with the kinetics of NHS-based labeling under optimal conditions, the tube format demonstrated a more linear response over the first 30 minutes, with signal leveling off at greater than 45 minutes. By virtue of its capacity for diafiltration, the Amicon Pro device permits fast and effective labeling in the presence of extremely high dye inputs, obviating the need for reaction optimization. Assessing the comparative workflows, the Amicon Pro device reduced overall processing time to 30 minutes. The main contributor in the tube format was replacement of the kit's 2 recommended overnight dialyses with 15-minute diafiltration spins. The device also demonstrated a significant improvement in antibody recovery. This most likely reflects sample containment within a single device for the entire process. The kit required multiple transfers between different devices, each with a potential for sample loss. For commercial antibodies, sample loss can be cost prohibitive, as well as limiting to the number of downstream applications that can be evaluated with a given batch of labeled antibodies. Functional performance of the conjugates was further evaluated by Western blotting. On the left, blots containing A431 cell lysate fractions were probed with the following anti-GAPDH conjugates: a commercial antibody, an antibody labeled according to the dye manufacturer's recommendations, and an antibody labeled using the Amicon Pro device. In addition to the FITSI labeling, shown below are examples labeled with Alexa 488, Alexa 647, and biotin using the Amicon Pro device. On the right, for each FITSI conjugate, the graph plots the signal strength for each band as determined by densitometry. From the dilution series depicted in Figure 5, it is apparent that for a similar amount of primary antibody, signal intensity was greater with user-labeled antibodies than a commercially available conjugate. This test also confirmed that there were no adverse effects of the probe's performance, that is, signal intensity, specificity, or background, incurred by using significantly greater amounts of dye, as was done with the Amicon Pro device. Overall, the device's centrifugal format, reduction in processing time, and greater retention lends itself to rapid, optimal labeling. Flow cytometry is commonly used for phenotypic analysis of cellular subpopulations and complex biological samples. The degree of fractionation permitted is dependent on the availability of antibody conjugates bearing spectrally distinguishable fluorescent tags, reliable determination of distinct subsets, is further dependent upon highly specific bright antibodies offering a significant difference in mean fluorescent signal intensity between positive and negative fractions. To demonstrate the utility of the diafiltration-based antibody labeling method for flow-based applications, 3 antibodies, anti-CD3, CD4, and CD8, were labeled with unique small molecules using the Amicon Pro device. Staining performance in human whole blood with the labeled antibodies was compared to that of commercial conjugates using equivalent antibody load. On the left, the representative dot plots depict identification of the PBMC fraction based on differential forward versus side scatter signal, further fractionation of human T cells, and total PBMCs was performed on the basis of CD4, CD8, positive expression. Cells in the upper and lower panel were stained using commercial and Amicon Pro labeled antibodies respectively. The table on the right shows comparative data for 3 samples stained with commercial antibodies and Amicon Pro labeled antibodies. As you can see, there was very little difference in performance as determined by MFI or population frequency. In summary, while antibody labeling kits are available with a range of detection moieties, the workflow is hampered by a requirement for multiple dialysis-based buffer exchange steps that are both time-consuming and subject to sample loss. Here we have demonstrated that all phases of the conjugation workflow can be easily and effectively performed within the Amicon Pro device. Specifically, when compared to other common buffer exchange methodologies, the centrifugal diafiltration method offered superior performance as measured by 4 key parameters. As the device was originally designed for resin-based affinity purification, it also provides a platform for upfront antibody purification or albumin carrier removal. Most significantly, by exploiting the rapid kinetics of labeling reactions, the process of continuous diafiltration minimizes reaction time and long exposure to excess dye, guaranteeing maximal target labeling while limiting the risks associated with over-labeling. Overall, the device offered a simplified workflow with reduced processing time and hands-on requirements without sacrificing labeling efficiency, final yield, or conjugate performance. With that, I'd like to thank you all for your attention, and we will take questions.
All right, thank you so much, Dr. Cappione. So let's go ahead to the question and answer segment. So if you haven't yet submitted a question for Dr. Bushnell or for Dr. Cappione, now is the right time to do so by clicking on the Ask a Question box at the bottom of your screen. So let's go ahead and proceed with the questions that have come in so far. So either of you who want to jump in to answer the question, feel free to do so. The first question is, how do you actually optimize a polychromatic flow cytometry panel?
No, that's a great question, Gwen. Optimization of a polychromatic panel involves several steps. The first of which is optimizing the antibody concentration by titration to ensure that you're getting the best signal to noise and best staining for each of the antibodies that you're going to use. Once you've optimized that, you would then go and optimize the voltage that you'd want to use. And this is a similar process to optimizing by titration, where you would use the optimized antibody concentration and then run a voltage series. Most of the time, you're not going to find a better voltage, but sometimes, depending on your instrument and depending on how the PMTs have been previously characterized, you might find a better voltage that gives you a good signal to noise, especially if you're using some of the newer reagents that have come out, such as the Brilliant Violet and the Brilliant UV dyes, which are not found in our beads that we use to characterize our instruments.
All right. Next question. It says, my antibody is in a hybridoma supernatant. Can I label this directly in the supernatant?
Hi, that's a good question, Gwen. I would recommend not labeling it directly in the hybridoma supernatant because of all of the other protein content that is in there will basically act as a sink and basically siphon away a lot of the fluorescent dye that you're using. Now, as I kind of walked through in our the protocol with the Amicon Pro, due to its ability to do affinity purification of proteins, you could actually remove your antibody via protein A or protein G resin up front within the Amicon Pro, elute it under your standard elution methodologies down into the Amicon Ultra component, and then move forward with your labeling of the antibody within the Amicon Pro, all containing it within a single device.
Okay, on to the next question. Can you give an example of how fluorochrome size would matter in panel design?
Yeah, that's a cool experiment actually that we were doing, looking at two nuclear antibodies, and we had one labeled with FITC and the other with PE. After stimulation of cells, we could find the FITC reagent looked beautiful, but the PE reagent didn't look so good. Our first thought was, oh, maybe there's an issue, so we swapped out the reagents. And so the FITC still looked good with the new reagent. The PE looked bad. It was only at that point we actually really started to think about the fact that phycoerythrin is actually a protein, and it's actually pretty big, 250,000 Daltons, I think, is the size. So now you're trying to get a very large protein attached to an antibody through the cellular membrane and through the nuclear membrane. So if you're doing intracellular targeted staining, smaller fluorochromes like FITC, like Alexa 488, Alexa 647, Cy5, Cy3, things like that, the small organic dyes are much better than the big protein dyes. You get them in easier, and you effectively have better staining.
Okay. Next question that's come in says, are there limitations regarding the kind of dyes or labeling reactions that can be used in the Amicon device?
Yes, there are. Due to the nature of how we actually clean up the labeling reaction to remove the unbound dye at the end of the process, there's a size limitation. So for the most part, the Amicon Pro method is limited to small molecule dyes, 50 dyes, Alexas, biotin, anything that's a small molecule. We'd not be able to accommodate labeling with something like PE or APC or any of the larger tandem dyes due to the fact that we use size-based exclusion for the cleanup step at the end.
Okay. Next question also has to do with the Amicon. It says, what amount of antibody can be labeled in the Amicon device?
So the Amicon Pro application is really limited, sort of small-scale antibody labeling application. We're looking at, we've worked with anywhere from 25 micrograms up to 200 micrograms. It's really based on the limitation of the Amicon Ultra 0.5, which is the buffer exchange and concentration component of the device. The limitation there is roughly 200 micrograms of protein can be held without fouling or causing issue within the device, lack of loss of performance. So it's really 200 micrograms is about the maximum that could be labeled.
Okay. Next question is, how do you design a panel using a fluorochrome that might contribute a lot of error to the panel?
So when you have a fluorochrome that is going to contribute a lot of error to a panel, you want to use it in a way that it actually is ultimately not going to be a problem. So the idea being that using it as a dump channel. So if I'm staining for T cells, I'll create a dump channel that has things like, that has markers for B cells or macrophages. And I would use that fluorochrome that contributes a lot of error to the panel, because when I exclude those cells in my analysis, my cells of interest will have none of that fluorochrome, so will not impact or be impacted downstream in my analysis. So I use it for things that I'm going to eliminate rather than I want to make a good measure on.
Okay, well, we are right up on the hour now, so we are out of time, and I'm going to wrap up the question and answer session now. But I would like to tell you that today's webinar has been recorded, and it will be available for viewing in the next few days. We'll send you an email with details on how to access the recorded webinar, along with instructions on how to personalize and print a certificate of attendance, as well as download a PDF of the slides. So on behalf of today's speakers, Dr. Timothy Bushnell and Dr. Amedeo Cappione, and our sponsor, MilliporeSigma, we sincerely appreciate your attending today's webinar, and we look forward to your attendance at future events from Current Protocols.